Hello guys, welcome to Bio 112. We are starting off Unit 1 with our Introduction to Biology unit. So first we're going to start off with Concept 1, which is just talking about scientific investigation and going about the difference in science and other fields of study and what makes science science. So starting off, talking about the difference between a fact versus a principle. Sometimes those words are used interchangeably, but they're not the same. So a fact is an objective, verifiable observation. So for example, water boils at 100 degrees Celsius. That's not an opinion, that is something that we can actually double check, make sure that that is what it is. And where a principle is a statement that's based on repeated experimental observations that describes an aspect of the world. So this is where it's based upon observations and it's based upon experiments, and its purpose is to describe something, how something is occurring, but it's not factually based as this is what it is all the time, mathematically, etc. Then you have the difference between a law and a theory. So a law is a very broad concept or principle. It's a how that describes patterns of nature, and it's often accepted as a fact. So for example, Newton's laws of motion. Something in motion is going to stay in motion until another force acts upon it. So it's something that we once again can verify and make sure that it is true and that it's always consistent. To whereas a theory is an explanation of things that we see, observed phenomena, that organize facts, and it explains why that's happening. So it never becomes a law because it's not explaining how, but it's explaining why that happens. We need to be very careful because when we talk about theories in the English language, a lot of times we talk about that as though it is a guess. And in science, a theory is more than a guess. It is actually based upon observations, and theories do change whenever we gather more information. So for example, the theory of evolution. It's a theory because you're never going to be able to observe the how it's happening in time. However, it is based upon principles and facts that we do see, and that is the best way that we can string it together with the information that we have at this time. So we know that science is based upon experimentation. That's why scientific findings are always changing, because new findings equaled updated conclusions. So in our lifetimes, there's been times where, especially like with health science, that things have gone back and forth. Peanut butter's not good for you, peanut butter's good for you, and it's based upon new research and new studies. So there's not one way to design an experiment. However, the investigative process tends to follow a general sequence. So typically we ask a question, what's going on? We conduct some background research to see what other people have done about it. We make a hypothesis, so kind of like our educated guess about what we think is going to happen. Then we test our hypothesis with an experiment, and then we take that experiment's data and analyze it and then draw conclusions from that data and then communicate them normally through a research paper. So looking at asking a question. So questions are often arise from the observations that we make. So first we need to define what an observation is. So it's a description of something that I can see, I can smell, I can touch, hear. This is not an opinion. It is a verifiable fact because we can see it or touch it or smell it or hear it. So for example, the ground is wet. Nobody is going to, need to fact check me on that because we both can see it. We both can touch the ground and feel that it is wet. We can make an inference based on our observations, and that's a guess about how that object or outcome happened based on our observations. So if my observation was the ground is wet, then I can make several inferences off of that one observation. I can make the inference of it must have just rained, or somebody dumped their Coke out onto the ground, or a fire hydrant is leaking, or a water pipe is leaking. So there are inferences, multiple inferences that I can make based off the observation of the ground is wet. When I'm looking at data and when I'm looking at observations, those data or those observations can either be qualitative or quantitative. So qualitative describes a quality, so it's kind of in the name. This is going to be a descriptive term. So for example, that is a green liquid. That is a green or a sweet scent. It has an adjective about it. Whereas quantitative, it has quantity in it. So this is using numbers to measure something. For example, that is 15 centimeters long. That organism has 8 legs. So it's actually giving numerical answers. Quantitative data can be accurate and or precise. So let's look at what those things mean. Accuracy is how close your measurement is to the correct or the accepted value. So is the data correct? Whereas precision is how close your measurements are to each other. Is the data consistent? Is it specific? So this is a really good visualization to be able to tell you the difference between precision and accuracy. Remember, precision means that you are consistent. So anytime where I have hit the target in the same location more than once, it is precise. However, if I am not hitting the middle of the target, I'm not accurate. So in the top left-hand corner, I am precise and accurate because not only do I have a grouping, am I consistent, but I am also hitting the accepted value, which is the bullseye here. So once I ask a question based off of my observations, I conduct background research. So this leads to defining the purpose or objective for my experiment. I'm trying to figure out the overall goal of a scientific investigation, which is to answer my question. And our observations led us to having questions, which leads us to taking research about our questions. What's already been found out about this question? And that helps me to define my purpose. So is my purpose to re-verify something that somebody else has already done, or is it to expound upon that? Once I've done my research, I construct a hypothesis. So this is, what do you think will happen? It's a testable prediction based on observations. So once again, it's more than a guess because it's based on your observations and it's based upon research. It describes a cause and effect relationship between variables. And the typical format for a hypothesis is an if-then. If something about the independent variable changes, then something about the dependent variable is going to change in response to that. So let's talk about what those variables mean. The independent variable is what an experimenter will be deliberately changing or manipulating in the investigation. On a graph, this typically is on the x-axis, and it should be the only thing that is different between the experimental groups. So this is what I am changing to cause something else to change. So for example, if I am trying to give a dog a weight loss, or if I'm trying to get my dogs to weigh less and I'm going to test different brands and see if different brands will cause my dogs to lose weight, I am changing the brand of food that I am giving them. So the independent variable would be the brand of food. The dependent variable is what changes in response to the independent variable. Normally this is going to be on the y-axis of the graph, and it's usually represented by the data you collect in an investigation. It is what is measured. So in that last dog weight loss experiment, the actual weight of my dog is what I am measuring that is in response to the food that they ate. So that would be the dependent variable. Whenever I am giving an experiment and doing an experiment, I want to write down a materials list, which is what am I going to need to conduct the experiment? I have to include amounts. I have to include brands. I want to be specific. And I write this as a bulleted list. Think about a recipe. You don't just put eggs. You have to give how many eggs. Are they small eggs, large eggs, jumbo eggs, duck eggs, quail eggs? You have to be specific. Otherwise, it's not replicable. It's not repeatable. Somebody can't look at it and do the same thing that you did precisely and accurately over again. I also have a procedures. Think about this as the instructions. So write out every step that was taken. I start with an action word, aka a verb. Place 5 seeds in cup. Pour 5 milliliters of water onto soil. And I need to include every step so that someone could replicate the experiment. You make this as a numbered list, right? So 1, do this. 2, do this. 3, do this. So you guys will get practice doing this in your experimentation, designing an experiment assignment. So please make sure to format your materials list and your procedures like it is designed here. So whenever I have an experiment, we said with the independent variable that it's the only thing that's going to change between my experimental groups. So what is an experimental group? An experimental group is a group that is being tested. It has the different independent variable options. Whereas my control group is my group that's used as a comparison with my experimental group. So it's the normal group. It either lacks the independent variable or it has the normal independent variable option. So let's go back to our dog food experiment. I don't want to not give my dogs any food. Okay, that could be a control group, but that also is probably going to kill them eventually if I'm not feeding them. So I don't want to do that. That's not the best option for this experiment. However, I can make my control group their normal food. So let's say that their normal food is Hill Science Diet. So that would be the control group. My experimental groups is all the other different brands like Iams and Royal Canin and Puppy Chow. So all of these different options that aren't their normal one. Like we said, the independent variable is the only thing that we want to change. That means everything else needs to remain a constant. So a constant is the aspects of an experiment that are held constant or consistent. I want to do this because it ensures that all the aspects of the trials are identical except for the independent variable. I need to walk my dogs the same amount no matter what food I'm feeding them. I need to make sure they get the same exercise, amount of sleep, etc. Because if I don't, then any difference that's measured in the dependent variable could be caused by something else that's not the independent variable. So then I can't be sure that one food is actually the cause of the weight loss or not. I want to repeat my trials. So this helps to ensure that the results aren't due to chance. It helps eliminate errors, and it helps ensure that my data is precise because I'm getting multiple readings, multiple sets of data that I can compare to one another. So once I've gotten my data, I can analyze the data. I look at my results. So I'm going to collect my data. I'm going to put it in an organized form, usually a table, and then I can actually make it into a graph that helps me present data in an easy to read way. Once I have my data in my graph and my table, I can analyze it. So I can highlight any trends or patterns that are seen in the data. So maybe one of the dog foods has a significant weight loss. One of the dog foods might increase my dog's weight. And then I can draw conclusions and communicate those. So I can either accept or reject my hypothesis. So data is going to either support my prediction or it's going to fail to support, aka reject my prediction. So if I thought that Iams, if my dog eats Iams dog food, then their weight will decrease. If that's what the data shows, then I can accept my hypothesis. But if actually Purina caused my dogs to lose weight, then I would reject my hypothesis. Data does not prove or disprove because it's not a law yet. It's not something that we can prove with 100% accuracy. So we don't want to say prove or disprove. And then with our conclusions and communication, we want to describe any of our real-world applications or uses for the information learned from the investigation. So maybe I talk about utilizing this particular dog food for dogs that are obese. So now that we know the scientific process, a lot of times science and technology are kind of lumped together in a broad overarching term, but there's actually a difference between science, technology, and engineering. So science is the advancement of knowledge, so specifically knowledge. I'm going to answer questions based on my observations via experiments. Technology is applying science to meet human needs and goals through the development of products and processes. So this is going to be solving a problem based on our needs. So for example, a cell phone is technology because it solves the problem of long distance communication. However, we had to learn and get knowledge about waves, like sound waves, and battery technology, and everything else in order to build that cell phone. And then finally, engineering is going to be applying science and mathematic principles to solve the problems. So technology is achieved through engineering. Whenever I am designing technology, I'm going to go through kind of this 4-step process. I'm going to identify my problem, so I'm going to clearly define my problem or need. I'm having a problem with fuel efficiency in my car. Solution design. I'm going to brainstorm. I'm going to research. I'm going to make sketches. I'm going to narrow down my best design for my product or my process. And I'm going to take into account my constraints. So it might be time, it might be cost. Maybe I have this really cool thing that gets me 500 miles to a gallon, but it costs $1 million. That's not going to be applicable to a whole lot of people, so it might not fit within my constraint. Then I'm going to implement my solution. I'm going to build it and I'm going to test it, and I'm going to make improvements to it until it works flawlessly. And then finally, I'm going to evaluate it. I'm going to determine if I solved the problem and if I met the constraints of the project. So maybe I made this technology device that we can put on cars to give them up to 100 miles a gallon. But the materials that I need for it are very rare. So it's only going to be able to be used in a few cars. That probably isn't going to be the best solution design because of limited materials.